Good morning, children. Before we understand megaspore and megasporangium, let us first discuss gynoecium. Gynoecium is the female reproductive organ in a flower. It can be studied under two headings, monocarpillary and multicarpillary. When one pistil is present, then it is known as monocarpillary. Example, beans, mango, coconut. Multicarpillary, when many pistils are present. Now, there can be two conditions. When the pistils are all united, then it is known as syncarpous condition. An example is Papaver somniferum. When all the pistils are free, then it is known as apocarpous condition. And for learning, it will become easy if you see all the beautiful flowers like lotus, rose, michaelia. Now, carpel consists of three parts: stigma, style, and ovary. The broader portion at the top is known as the stigma, on which the pollen grains will land during pollination. This neck region is known as the style, through which the pollen tube will cross, and this swollen portion is known as ovary. Inside ovary, cavity is present, which is known as locule, and there is cushion-like structure, which is known as placenta, and this is ovule. So ovule will be attached with placenta with the help of a stalk known as funicle. The place where the funicle joins the ovule is known as hilum. Ovule is known as megasporangium. It is known as megasporangium because megaspores will develop from ovule only. Now let us understand the structure of ovule in detail. You know, after fertilization, ovule turns into seed. So the covering will turn into seed cover. So it is covered by integuments, and this integument is not continuous at all places. So the place where the integument is not continuous on the ovule is known as micropyle, and region just opposite to is known as chalaza. Inside the ovule, diploid cells are present, which is known as nucellus. Not all the cells, but only one cell towards the micropillar end will behave as megaspore mother cell. So, in megaspore mother cell, which is diploid in condition, one meiosis will take place. And you know, meiosis 1, meiosis 2, there are two phases in one meiosis. So, after that, 4 haploid cells will be formed, which will be known as megaspore. 3 of it will degenerate, only one will remain as functional megaspore, and therefore it is known as monosporic development. Now, there is a difference in this megaspore and microspore. What? In microsporangium, all the sporogenous tissues, they behaved as pollen mother cell. But here only one towards the micropillar end behaves as a megaspore mother cell. Hope I have made the difference clear.